Okay, so welcome to this next video on phosphodiesterase enzymes. Okay, so I've told you that there are 11 different families of phosphodiesterase enzymes, and within each family there are multiple different genes which actually code for phosphodiesterase enzymes. Now, two of the families that we're going to look at in detail, because they're actually noteworthy, are phosphodiesterase 1 and phosphodiesterase 4. So we're going to look at these two. Now, phosphodiesterase 5 is very, very famous because phosphodiesterase 5s are the targets of the famous drug sildenafil, more commonly known by its infamous brand name as Viagra. But phosphodiesterase 5s aren't particularly specific to cyclic AMP. They're more specific, in fact, to cyclic GMP. And since this is a playlist on cyclic AMP signaling, we're not interested in phosphodiesterase 5. Relevant to cyclic AMP, particularly relevant to cyclic AMP, are phosphodiesterase 1 and phosphodiesterase 4 families. So I've told you that phosphodiesterase 1 family consists of 3 genes. So how many genes are in phosphodiesterase 4? Well, basically, in the phosphodiesterase 4 family, there are 4 different genes which all code for enzymes which are similar enough to be considered in the same family. So there's 4 different phosphodiesterase 4 enzymes, and basically they are denoted phosphodiesterase 4A. So you have 4A. Then you have phosphodiesterase 4B, and then it goes on. You go on 4C and 4D as well. So you'll have 4C and 4D, and those are the actually different enzymes. So those are encoded for by different genes, and they will have slightly different amino acid sequences. Now, phosphodiesterase 4s are all inhibited by a very an important drug, which is rolapram. So they are all inhibited by rolapram. Okay, and another important thing is to talk about there's more variability even than this. So we've got 11 different families, and all these families have loads of different genes all coding for phosphodiesterase enzymes. So you might think that's a lot of variability already, but there's actually another layer of variability because there are different ways of splicing each one of these enzymes. So let's say we have our gene over here. So this is our gene. The gene codes for RNA, basically. So the gene is going to be transcribed. So let's say this is our phosphodiesterase gene, PDE gene. Basically, the gene is going to be transcribed and turned into mRNA. So here's our piece of mRNA. And initially, the mRNA will just be a complementary strand to the entire coding strand of the DNA. So there are 2 strands of DNA that are complementary, and basically one of them will be used to make the mRNA, and you will just make your piece of mRNA so that it's complementary to that coding strand. So this is known as pre-mRNA. However, this piece of pre-mRNA will contain loads of non-coding portions. So the gene contains portions that are actually going to be used to make phosphodiesterase, and then portions which aren't actually going to be used to make phosphodiesterase. The portions which are actually used are known as exons. So let's say these are the exons. I'll mark them more visibly. So these are the exons here. And then there are portions in between which aren't actually used in the production of the protein, and those are introns. And basically what you have to do is, when you've created this pre-mRNA, it's just a copy of this. So it contains all the introns, and it also contains all the exons. I should have probably said that the other way. It contains all the exons, and then it also contains all the introns. So what you need to do is remove the introns, and that process basically is known as splicing. But when you splice, there are different ways basically of splicing. You might have some exons where there's actually a choice of which one to put in, i.e. you could say have 2 exons here. And actually, to make a functional phosphodiesterase enzyme, you can't put in both of those exons, and you might have a choice of which one to put in. You can put in one, or you can put in the other, and that will make slight variants of the enzyme, basically. So there's basically splice variants as well. So when you undergo the splicing process, you can get different types of mature mRNA pieces from the same gene, basically. And it's through this sort of choice of which exons to use. I mean, some of the exons will be essential. There won't be, if you don't use them, you don't get a functional enzyme. But some of the exons, there will be a choice of whether you put this one in or you put this one in, basically. You'll have to put one of them in, but you can choose which of them to put in. And they'll obviously have different sequences of organic bases, and they'll code for different sequences of amino acids. So you'll get slightly different enzymes, basically. So there's also basically different splice variants of these, each one of these genes. So they give off different splice variants as well. So I'll show it like this. Different splice variants. Okay, so you have 11 families of phosphodiesterases. Each of these contains multiple genes that code for phosphodiesterase enzymes, and each of the genes has multiple splice variants. So there are a lot of different types of phosphodiesterases. Now, the important thing about phosphodiesterase 4s are that they are activated by protein kinase A. So they break down cyclic AMP and they're activated. They will start breaking down protein cyclic AMP in response to being phosphorylated by protein kinase A. Phosphodiesterase 1s are also selective for cyclic AMP, so they're also very interesting for us, but they aren't activated by protein kinase A. They are instead activated by calcium calmodulin complexes. So when calcium goes up, it will bind to calmodulin, and the calcium calmodulin complex will then activate phosphodiesterase 1. Okay, and we'll see more about these in upcoming videos.